Methodical. To start, let's review the basic structure of a feed-forward neural network. In this system, signals travel in exactly one specified direction, moving from the input layer straight through the network and out to the prediction layer. When one of these networks contains one or more hidden layers between the input and output, we classify it as a multi-layer perceptron, or MLP. But using a standard MLP for high-resolution image processing introduces a major structural bottleneck. The sheer volume of data in a modern image causes the network's densely packed connections to physically overload the system's memory capacity. To bypass this mathematical roadblock, we have to abandon the idea of connecting every node to every other node. That structural shift gives us the convolutional neural network. Let's calculate exactly why an MLP fails here. A standard 256 by 256 pixel color image has 3 color channels. That requires an initial input layer of exactly 196,608 individual values. This animated diagram demonstrates the problem. When we attempt to densely connect all 196,608 inputs to the next hidden layer in an MLP, we immediately generate tens of millions of distinct weights. Processing tens of millions of weights for a single, relatively small image requires massive memory allocation, causing standard hardware to simply stall out and fail. A convolutional neural network solves this by enforcing sparse local connections. Instead of every neuron looking at the entire image, they only look at very small, specific sections. We accomplish this through a mathematical operation called convolution. As you can see in this diagram, we use a single 2D filter square that glides step by step across the larger image matrix, creating a new mapped feature at each step. Because of this sliding window, each individual neuron in the CNN is restricted to processing just one local patch of the input data at a time. Shifting from a dense global web to a localized sliding window drastically reduces the computational load, and it perfectly preserves the spatial relationships of the pixels in the image. This concept of analyzing small, local patterns is not strictly a mathematical invention. It directly stems from biology, specifically how the animal visual cortex operates. Researchers Hubel and Wiesel discovered that certain neurons, called simple cells, react only to basic local illumination, like small spots or straight edges. They also observed complex cells. These ignore simple local patterns entirely, firing only when they detect larger, continuous shapes, like a long line of light. The biology operates in a strict hierarchy. A group of simple cells act as edge detectors, summing up their individual responses and feeding that combined signal forward into a single complex cell. This biological hierarchy maps directly onto the artificial layers of a CNN. Early layers scan for edges and spots and feed those signals into deeper layers equipped to recognize whole shapes. By building complex shape recognition from simple, local observations, convolutional neural networks algorithmically mimic the way humans and animals process visual data. We can see these concepts form a complete system by looking at the first successful convolutional neural network. Created by Dr. Jan LeCun in 1998, it was called LeNet. Looking at this architectural blueprint, the initial stage of LeNet consists of 2 distinct sets of operations, a convolutional layer followed immediately by a pooling layer. After the multidimensional image data passes through those initial sets, it hits a transition phase. Here, a flattening layer takes the 2D data and compresses it into a single flat 1D sequence. That flat sequence then feeds into the final stage, 2 fully connected layers, terminating in a softmax classifier, which outputs the exact probability for each possible label. Note one historical discrepancy. In the original 1998 paper, LeCun used a specific type of learnable average pooling. Today, the parameters in average pooling layers are static and no longer learnable. While individual operations like pooling have evolved over time, this sequential block-by-block blueprint established the permanent structural foundation used in modern image recognition. To prove this architecture worked, LeNet was trained and tested on the MNIST dataset. This dataset is a collection of 70,000 black and white images, each containing a single handwritten digit ranging from 0 to 9. The researchers split the data rigorously. They dedicated 60,000 images entirely to training the model. reserving the remaining 10,000 strictly for evaluation. The network's ultimate objective was straightforward: take a raw input image from the evaluation set and accurately predict its numerical label. By successfully mapping 70,000 human-written digits into accurate classifications, Linnet proved that machines could reliably interpret complex visual variations using convolutional mapping. Replacing densely packed connections with a localized sliding window solved the massive parameter bottleneck that stalled earlier networks. Stacking those localized layers sequentially mirrored the biological hierarchy of simple and complex cells found in the visual cortex. And by combining convolutional feature extraction with flat, fully connected layers, Linnet successfully categorized the entire MNIST dataset. This blueprint maps the entire theoretical pipeline, from the raw input matrix to the final softmax probabilities. Understanding this specific structural sequence is the required first step to successfully implementing a convolutional neural network in code.